let's start with a quick introduction. Do you want to give a quick introduction?
Yeah, I'm Andrew Gray. I'm the founder and CEO of Karma Biotechnologies, and we are developing vaccines for autoimmune diseases, starting with MS and myasthenia gravis.
Very cool. So what's the story? How did you end up with this idea?
Well, this story, I'm 41 years old, and this journey started 33 years ago. So it's been a while. When I was 8, my mom was diagnosed with myasthenia gravis. And it was this really seminal moment in our family's history, right? Where I remember this doctor sitting me down and telling me, we've worked out why your mother has all these weird symptoms. And it's this disease. And he said myasthenia gravis, which of course I didn't understand because I was 8. And he said, you should expect that she won't be around in a couple of years, right? And that was mind-blowing to young me, because the thought I have now is no awards for bedside manner for that guy, first of all. Second of all, I couldn't understand why they couldn't help her. They were telling me there was no cure for myasthenia gravis. And I'd grown up on Star Trek, right? And Dr. McCoy always figured it out. He'd always come up with a cure at the last minute. And and I couldn't understand why this guy in a white coat was telling me, there's nothing we can do. Get used to it. And in hindsight, that really triggered my interest in science and biology specifically. And everything I've done in my career has really been targeted towards, okay, how do we understand the immune system? How does it work? Why does it fail? What can we do about that to solve the problem? And, that my whole career has been taking one step at a time towards that goal. And Karma is really the culmination of that. One thing I do get asked is, people say, oh, sorry about your mom. That must have been really hard. She's actually very much still with us. She is incredibly stubborn. And she's one of the lucky few, though, who've lived this long with a really horrible disease, but thanks to COVID being basically a shut-in for most of this year. Right. So, yeah, it's not an easy life, that's for sure.
Yeah. So if we go back, did you go for PhD? What's your science story before the entrepreneurial story?
Right. So I went to the University of Edinburgh to do my undergrad. Mm-hmm. I actually did genetics rather than immunology as such, because I wanted to understand things at a more fundamental level. So went to Edinburgh for undergrad, was doing this genetics degree. And in my second year, I was given this opportunity to go to Berkeley for a year as an exchange student. Mm-hmm. And I really, I looked at the weather and I thought, what? A year in California would actually be very, very nice. So I would really love to have a year in California. So I went to Cal and I spent a really good year there, made a lot of friends, got to know the Bay Area a little bit, and I knew I wanted to come back. So I applied for PhD programs in the US, focused on California, but all over the place. And I got into USC, which a problem as an international student is that a lot of PhD programs are NIH funded and they can't really take international students, which is fair enough. It's all US taxpayer money. USC, of course, is a private institution. They can take whomever they like. So I got a spot at USC to join the PIBS program, which is programs in biomedical and biological sciences. Joined the lab of a guy called Martin Kast, who is this very famous tumor immunology guy who developed all these different cancer vaccines. He's very famous in the HPV scene. And the reason I joined his lab was because he is a very good pitchman, and he told me that this was the right lab to join, because the problem at the time, this is 15 years ago with immuno-oncology, was you could vaccinate mice, for example, and they would not grow a tumor. And everyone's excited about that. But that doesn't really help a person who already has cancer. And when you vaccinate mice with cancer, 9 times out of 10, nothing happens. And it almost never translates to humans. So there was this known problem in tumor immunology that the tumor fights back. And it was thought at the time, cancers have this way of evading the immune system, right? Tumor immune evasion is the term. Nobody really understood it. And Martin, when I was interviewing, you're kind of interviewing each other when it comes to picking a PhD mentor, right? And he was pitching me on his lab as much as I was pitching him on me joining. And he was like, my lab really needs to understand how tumors do this. How do they dodge the immune system? what are the cellular and molecular mechanisms of that? If you understand that, you'll understand how you can use those mechanisms to restore immune tolerance and then solve autoimmunity. And then you can have your career in autoimmunity, and, we'll be friends and not competitors when you're a big boy. So I bought that argument, and he was absolutely right. And sure enough, most of what we know about immune tolerance now, or a lot of what we know about immune tolerance, it directly comes out of the immuno-oncology space and figuring out how it is that cancers avoid the immune system. And the truth is, they don't just dodge the immune system. They don't just hide from it like we used to think. They actively subvert it. It's really insidious. So you'll get these immune cells that are tumor specific, and they'll go into the tumor, they'll infiltrate, and they'll start doing their thing. And the tumors that survive not only switch them off or blunt their effectiveness, they actively flip them to be tolerogenic, so that they will actually tolerate the tumor and protect it from other immune systems that come in. And that's what my work was on during the PhD. Regulatory T cells were just really formally discovered at that time, and I focused my efforts on it. So yeah, everything I know about immune tolerance comes from the tumor immune microenvironment.
Right. So it is also about protecting the immune system from cancer cells. Did you do any work, like how you can really empower or prepare immune cells to fight back and win the battle with tumor cells?
Yeah, so that was more a follow-on from my work. So in my last couple of years, my project was followed on by this very talented new PhD student called Lisa Yan, and her work was, how do you modify the tumor microenvironment so it's less immune suppressive and overcome that tumor microenvironment that's so immune suppressive. So she figured out a way to engineer the immune cells themselves so that they gave out a more positive signal instead of a negative signal to the immune system. And that greatly improved things. I was very excited.
That's pretty cool. I was reading somewhere, and it was talking about how the future is all about leveraging internal capabilities, empowering immune cells, empowering microbiome. So there's also a field called, what they call it, oncobiome. It's like immuno-oncology plus microbiome. So leveraging internal strengths, empowering those internal capabilities to win the battle, basically, whether it is against cancer or whether it is against infectious disease.
Yeah, whatever it is. I think that's true. Certainly the move now is more towards immuno-oncology, and how can we, the great success has been with these immune checkpoint inhibitors, right? Where you're effectively taking the brakes off the immune system, and the tumors are very good at putting those brakes on, right? Like they'll express molecules that are essentially the brakes of the immune system. And a lot of these drugs like Keytruda, for example, they're actually antibodies that will bind to those receptors that are putting the brakes on and prevent that from happening. So you unleash the immune system, but it's non-specific. It's basically you're taking off the brakes and just letting them do their thing. And I think that the next generation will be pairing that with cancer vaccines that do induce an antigen-specific response. And that's how you can go from, Keytruda, I don't know what the current success rate is with Keytruda in the various combinations that you see in the clinic now. But say it's 20% success rate of these tumors disappear, right? Which is phenomenal. And 10 years ago, if you had stage 4 melanoma, you were done. And now with Keytruda and Opdivo, things like that, you can survive a lot longer. Or even in the most extreme examples, you'll have patients whose tumors literally vanish in weeks, which is phenomenal. But then what do you do for the other 80% of people? And then you need, something that you need to train the troops to go in to that tumor specifically and take it out. And that's where the cancer vaccines come in. It's a very exciting time. And I think generally immune engineering, the time is coming where we can engineer the immune cells, right? We can engineer immune cells to do what we want. CAR T cells are a great example of that, where you literally engineer the patient's own T cells with a new T cell receptor that is specific for some tumor antigen and put them back in and let them do their thing. And that's amazing. But it's really complicated and very, very expensive because you have to take these cells out of the patient, and that patient is really sick with cancer, send them to some lab somewhere, keep everything sterile, engineer those T cells, grow them up so there's billions of them, send them back to the patient and reinfuse them a few weeks later. And that's a heavy lift, and it's doable. And companies like Kite Pharma, for example, who were born a few miles from where I am now in Santa Monica, did a phenomenal job of making that real. Everybody thought there's no way you can do that logistically. It's too hard, too expensive, it'll never work. They made it work, right? And hats off. And that's why they're acquired for $12 billion or whatever it was. But I think the future is, and I'm not the first person to think this, I think the future is doing that kind of engineering in vivo. So you can go to your doctor and get a shot that will engineer your T cells inside your body. And that process then is weeks shorter than it would be. And also, less risk, less expensive. It's the wave of the future. Nobody wants to do ex vivo long term. I think even the companies that are doing it well don't want to do it long term. And that's really what Karma is all about, is figuring out what can we do to engineer the immune system inside the patient's body and give it the tools to fight whatever the disease is. So basically, your immune system sees the world in black and white. There's either self or not self. And if you're not self, you get attacked. If you're self, you get tolerated. And you would think that, okay, well, those are clear rules, that's fine. But there's always a gray area, right? Every time in immunology. And occasionally, something that is self is mistaken for not self. Right. And then you get an autoimmune disease. And the flip side is, in cancer is close enough to self to convince the immune system, hey, I'm you. Don't attack me, right? I'm one of you. I'm a good guy. Leave me alone. But it's not self. Right. So being able to retrain the immune system to those subtle nuances, that's the future, right? And if you can convince the immune system, actually, that tumor, it's not self, not really. Go and kill it. And the flip side is, if, say, a patient has rheumatoid arthritis and the collagen in their joints is being attacked, you could retrain the immune system and say, hey, look, that collagen that you're attacking, it's actually self. That needs to be back on the good guy list. Stop attacking that. And that's exactly what we developed our platform, which is, essentially vaccines to achieve that we call Zavines. That's what we designed that to do, to retrain the immune system so that it re-recognizes self components as self, so it stops attacking them and therefore blunt this autoimmune response.
That's awesome. And I want to go back to what you mentioned is more personalized, patient-specific treatment. So how it's going to work, even if you just talk about focus on Kite Pharma, like how does it work? Like, do they have to do some test for the patient, or do they have to recapitulate patient's condition on organ on a chip, like human on a chip, microfluidic chip?
No, I don't think so. I don't know Kite's secrets. So
talking like generic, like in the future, how do you think?
Well, with CAR T cells, the specificity comes from taking the patient's own cells and engineering them. And the reason you do that is because we're back to the self versus not self, right? So if you needed a kidney transplant, right, and I'm a kidney donor, we would need to do all this testing to make sure that we're a tissue match, right? Which is super unlikely because we're not related. But if my twin brother I don't have a twin brother, but if I had a hypothetical twin brother and he needed a kidney transplant, we'd be almost a perfect tissue match, right? And I'd be the best candidate. Same for my sister, right? My little sister, God forbid she ever needs a kidney transplant, I'd probably be the best possible donor. Well, with your CAR T cells, it's the same thing. You can't just take anybody's T cells and engineer them and put them into any other patient, because they'll immediately be recognized by the rest of that patient's immune system as not self, and they'll get attacked and won't be able to do their job. So to avoid that, you take self cells out, engineer them, and put those back into the same patient. And that's one of the reasons it's so expensive, because it's so specific. And you look at the control centers of where they manage this process, and it's like something out of NASA JPL, right, where they're tracking every patient at every step and making damn sure they know exactly who's where, what the status of their cells are, when it gets sent back. It's very, very complicated and a logistical nightmare, really. But you've got to do it, because it's the only way to guarantee that that patient gets their own cells back.
Right. So I think this is more of an obvious reason why you have to make it personalized, because there's a good chance of immune rejection. So if you engineer someone else's cells and then administer them, there will be rejection. But I still think not just for immune rejection reasons, in the future of biopharma, the future of drugs is more personalized. I was reading somewhere that, for example, liver cirrhosis is not one disease, few hundred type of disease, right? Because different bodies are different. So in the future, currently, because I don't know, over 10,000 diseases need a cure. So we have to take more blanket approach. But I think once we have things in our control, in our hands, once we have cures for most of the disease, industry will move towards more personalization. So you really need sequencing data, microbiome data, immune systems data to create a drug that really works.
Right. I think we'll see that level of personalization. Certainly, you're seeing it a little bit in tumor immunology or immuno-oncology. I think we'll need to see that also in autoimmunity. Right. So the big challenge in autoimmunity is, so say a person has MS, for example.
Right.
So in MS, the immune system is attacking the myelin sheaths of the nerves. And that's a bit like, if you imagine an electrical cable, it would be like stripping the insulation off the cable so that it, the signal can't get through properly, and you get a short circuit. That's the best analogy. Well, how the immune system does that is variable. So the immune system mostly recognizes proteins, and which the proteins in the myelin sheath are actually recognized and which parts of those proteins are recognized varies from patient to patient. And we know that there are some dominant ones that show up in a lot of patients, and that's what you target first. But there's a lot of people who don't, you can do a test to see which antigen's being attacked, and it comes back, everything comes back negative. And is that because you're just not detecting something that is there, it's a false negative? Or is it that patient, they're getting MS because their body is attacking a protein on the myelin sheath that is very rare and unusual? And for that patient, how do you create a vaccine for them if their immune system is attacking something so off the wall or rare? So I think we will have to move to testing each patient's T-cell repertoire and figuring out, okay, what exactly are you attacking? Because if we know that, then we can give you the right tolerogenic vaccine to solve that problem. So I agree with you. It's, but fortunately with, next gen sequencing, things like that, there are companies, there's a very good company that I really like. That's what they do. They sequence people's T-cell repertoires and tell you, this is what this person's T-cells are recognizing and probably attacking. And that's very exciting, very powerful, right?
Right. I wonder if we need to even reach that stage and introduce personalization in this industry at scale. We need a digital identity of biological data of every person, right? Mm-hmm. So if you have a patient, you should have access to their sequencing data, right? Mm-hmm. You should have access to their immune system data. You should have access to, I'm saying you, in this case, probably physician or researcher, access to microbiome data. So all these factors that really make a difference, how the body respond to a drug. You really want to understand that. Currently, we are just blindly shooting, right? So we create something that works for a good number of people. We introduce that to the market and hope it will work for everyone. But that's not the ideal way.
No, and it won't work, right? And it won't work for everyone. And in the cancer field, we've sort of accepted this, where a successful cancer drug trial is one that improves quality of life or longevity in some minority of patients. And for some people, it just does not. And up until now, you would just be like, well, too bad for them. Let's move them into the next clinical trial and hope that works for them. Well, I think now, certainly with things like machine learning and AI being what it is, I think we'll be able to much more efficiently parse out which patients are more likely to respond to a given drug, or which patients are, not responding because generally, like with cancer vaccines, for example, a lot of these people are very sick and their immune systems are shot, right? They're just completely run down. Are those people so sick that they would never have responded to any vaccine, or is it specifically this vaccine that they're not responding? And I think we can parse out that data. And to your point about having a patient's data, that's really important. And your genome basically won't change, but your T-cell repertoire changes all the time based on what you're exposed to. Your microbiome changes all the time depending on what you eat, whether you've had a course of antibiotics lately or whatever it is. And having some baseline in your electronic medical record of this was what this person's T-cell repertoire or B-cell repertoire looked like when they were 25 years old and healthy, and this is what their gut microbiome gave us back then. Having that baseline to work from, then when diseases show up, you can refer back to that and do sort of retrospective trials in potentially millions of patients and be able to pick out those patterns, like what did change in those patients.
Right.
What if, we see this spike of MS patients, and then, oh, by the way, it's much more likely that this particular species of pathogenic bacteria shows up in the gut of these people, and there's some correlation there. I'm making that up. It's, just an example.
Right.
But those patterns, you can't respond to what you don't measure. Right? You can't treat what you don't measure.
Right.
So if you have those data, but it's expensive, and who does that, right? Who pays for that? Is it the government? Is it insurance companies? Is it patients? People are more open to genetic. Everyone has 23andMe now, right?
Yeah.
Is that going to be useful later on? Maybe that's 23andMe's business model is.
Yeah, I do believe 23andMe, companies like 23andMe and Ancestry.com, and for Biome, what do you call it? Biome.com? Ubiome.
Yep. Ubiome, yep.
So all these personalized testing companies will play a huge role, but I would love to empower patients to control their data. Mm-hmm. So they don't just have their data, they can monetize it, because until you have that level of incentivization, you're not going to have a digital identity of every patient in the world or every human in the world. So we really need a new decentralized incentive system, because otherwise these companies like 23andMe will basically own the data, and there's no incentive for end user. Now I don't want to sound too Black Mirror-like or far-fetched, but when you were saying that when disease happen, we can go back and see what was their profile, whether it's biome profile or T-cell profile. But I think we can take one step further. Before even disease happen, if we can track the changes in real time, we can say this disease is going to happen if you don't stop that mess up, if you don't change your lifestyle, right? So
it's a different story. I don't know how many times my doctors told me you need to stop drinking so much coffee is the thing I get all the time. No coffee, no science. That is the general rule. But, so changing patient behavior is always very, very hard. But I do agree with you. It's stepwise, right? So if you do that retrospective study and you figure out, okay, there's correlation, then you can apply it forwards to a new group of patients and see, okay, these people, this group of people, whether it's a shared behavior or a genetic marker or combination of the two, more like they have that. They're staring down the barrel of this thing, right? They're heading towards diabetes or MS or cancer or whatever it is. At least then you can warn them and say, hey, look, you've got this marker, right? And, if it's easy enough for the patient to adopt, it will happen, right? So the use of statins, for example, is a really good idea.
Right.
Statins are very, very easy. They mostly have limited side effects. You pop a pill a day and it reduces your risk. And if you have, high cholesterol, and it runs in my family, right? So I'm sure I'm going to run into that at some point. Then, it's an easy modification, right? I think then things will be adopted. And if it's as simple as you get a test and it comes back and it says, oh yeah, your LDL is too high, go on statins and you'll reduce your risk, fine, right? And if we can get to that point with things like, autoimmunity and cancer and things like that, and say, okay, you are heading down this pathway. We want to immunize you against prostate cancer, say, right? And for that's an easy example, because basically all men, if you live long enough, you'll get prostate cancer, right? So part of my thesis actually was, I had this observation that if you vaccinated early enough in the progression of cancer, then you would basically prevent it forever. And the mice that we were using, they would die of old age. All the controls would be long gone. And these vaccinated mice that were genetically engineered to get prostate cancer never did. Actually, that isn't true. They did get it. But then when you looked at the prostates under a microscope, they had these little pockets of very high grade, very aggressive prostate cancer, and it was completely surrounded by immune cells. It was like the riot police had just surrounded the bad guys whenever they popped up and just, take them down. So that's really exciting, right? And that early intervention immunologically, as a guy who's over 40 now, and at 50 you have to start worrying about that stuff. I would, if it was available, I would take a preventive prostate cancer vaccine right now. Same for HPV vaccines, right? Classic example now that's sort of taken for granted, but 10 years ago it was science fiction. Or, 15 years ago. And that HPV infection that leads to cervical cancer, head and neck cancer, you can step in with a vaccine before those behaviors that lead to being infected by HPV kick in, right? So teenagers, and dramatically cut the rates of cervical and head and neck cancer in young adults now. Like the generation that got vaccinated, starting about 10 years ago, they will not have to face that. And it's horrible. It's cervical cancer in particular is a very, very, it's an awful disease, and it kills a lot of young women. And hopefully we've pretty much wiped that out, or will, once it's universally accepted to have your HPV vaccine when you're a teenager.
Right. And is there any, is it even possible? I'm sure everything is possible, but in, let's say, a 10-year period in the future, we can track immune profile, the fluctuations in immune profile in real time?
I think that's doable now, really. It's okay. So for, say, T-cell repertoire, because this is something, my friend's company does this, and, I've seen her pitch and all that, and I understand her work. They can tell you the sequences of all of the T cells that you have that are, and how relatively how many you have, which is an indication of which ones are responding to something and which ones aren't. You could do that today. So imagine if you had taken somebody's baseline T cell repertoire genetic profile, right? And you could tell, like they have, I'm oversimplifying, but say 1,000 T cell receptors that they're expressing. And then suddenly, so say you did that in summer of 2019, and then suddenly, and you're monitoring these patients every now and then. Suddenly in early 2020, you get this new T cell or B cell population that's coming up. It's like, what on earth are they responding to? Well, the answer would be COVID, right? And that would have given us an early warning of like, there's a new kid on the block that people are starting to respond to. We don't know what it is yet, but we need to get ready for that next pandemic because something's going down. And that would have been very powerful. But you have to have this longitudinal monitoring where somebody gives a blood draw every 6 months or a year or whatever it is. And you need very big patient populations to make that work, but it would be possible.
I'm thinking if there's wearable that can access blood, like a wearable device.
I mean, it's kind of the holy grail, right? But like you say, everything's possible. I don't think there's a practical way right now to do that, but it's like everything else. There was no practical way 20 odd years ago to sequence the human genome. And now you can have it done for $1,000 in a few days. And that's, certainly sequencing, the cost comes down all the time. It's like this reverse Moore's law thing. And, that's exciting. And it does empower where you can do these massive sequencing projects and get tons of data. The problem, I think, is actually not getting the sequencing anymore. I think it's how do you analyze it? Because no human being can analyze all of that data. Then you've got to have machine learning and the bioinformatics side becomes the big challenge. And that's something that I'm very interested in.
Yeah, like generating data is like not even half the battle, right? You have to really make sense out of it.
Yeah. Otherwise, it's all just noise.
I want to move to. I can talk about the science stuff for hours, but I do want to learn more about your entrepreneurial journey. How did you become an entrepreneur? What was your journey like? I'm trying to understand if someone listening to this recording for a week, month, and like aspiring bio entrepreneur, what advice you would give to that person? First, let's talk about your entrepreneurial journey, then we can talk about some advice.
Yeah, so I was coming into the last year of my PhD, and I was having that kind of classic end of PhD, what do I do next thing? And the choices are industry or try and be a professor in academia, which is pretty much a Ponzi scheme because there are almost no tenure track positions. And you have to come from a really rock star lab and have phenomenal papers and tons of grants. And, I'd done pretty well, but I wasn't being published in Nature.
Right.
So that path I didn't think was really an option for me anyway. So then I was thinking, well, do I want to go and work for a big company right out of grad school? And that didn't appeal to me either. And then the other options are things like scientific writing, consulting, and none of that appealed. So I was really struggling with, I love science. I love doing the science. I love solving problems, but I couldn't see where I could fit in and move the needle and actually do something. So, I'm struggling with this. And by complete coincidence, a professor called Tom, USC's Marshall School of Business. I don't know how he figured this out, but he realized that the health sciences campus at USC, which is its own little island on the other side of downtown from the rest of the campus, was full of all of these young, bright PhD biotech people who were developing all these amazing technologies, and nobody from the business school was talking to him. So he came over to our campus once a week for a semester on a Tuesday night, and he basically did this pop-up entrepreneurship class. And I think I missed the first one, and a friend of mine was like, "You've got to come to this, it's really cool." And as soon as I heard him talk, I was like, this is how you go from having an idea or a technology to figuring out your market to building a company. And he went through it step by step. This is generally how you do it. And it completely changed my world. And the light bulb went off. That's it. That's what I want to do. I want to build a company. And, I don't know why it felt so right, but it just did. And from then, I joined a lab as a postdoc, specifically because the PI wanted to spin out a technology. And that was the first company that I led for 3 years. And after that, I did Karma. We decided, the team that I worked with then, we wanted to stick together. We worked well together. And we sat down in a brewery one day and literally brainstormed with a notepad, the three of us sitting around this table having a couple of beers, and we were like, all right, what are we going to do next? What do we want to build? And that's where Karma was born. And we literally drew the blueprints of our technology in this notepad. And that was version, I don't want to call it version 1.0, it's more like version 0.1, but it all started there. And that was 4 years ago, basically almost 4 years ago to the day, actually, when we did that. And we founded Karma in March of 2017. And we've gone basically from strength to strength since then. It's been a hard road. So one thing we knew we didn't want to do was license technology out of a university. It's complicated and difficult. And, the universities have different incentives than the entrepreneurs. Right. It's very hard to come to an agreement that works for everyone, especially when you're a tiny little fledgling startup and you don't have much cash, and you definitely don't have the kind of cash to license technologies from universities. So we decided with Karma from day one, we're going to build everything in-house until we hit a certain size, because we knew we had the skills between us. So I was the immunologist, cell biology guy, and my two co-founders were a material scientist, Don Johnson, who is just genius. He's at BMS now. So he's a phenomenal scientist. And, I'd known him basically, for the first company, I hired him right out of grad school. He was amazing. I knew I wanted to work with Don forever. And then the third co-founder is a chemist, Alan Johnson, no relation. And I'd known him for 15 years playing rugby out of Pasadena Rugby Club. And, between those three skill sets, we knew we could build something really cool. And it was a case of, okay, what do we do? And the concept for Karma originally was, we knew there was this pain point in cell and gene therapy, which is how do you deliver your gene therapies to cells without using viruses, which have all kinds of problems. The two big ones being, it's hard to scale up viral production. Second problem is your immune system is very good at recognizing viruses. So usually, if you're giving a gene therapy with a viral delivery system, you get 1 or 2 shots at that before your immune system starts neutralizing the virus, and then you can't use it anymore. And that patient can't have any more treatments. There are ways of engineering the viruses to avoid that, but it's very, very difficult. So we thought, okay, how can we do this? And we all had this nanoparticle experience. So we were like, we can replace that using a nanoparticle. And lipid nanoparticles to deliver genetic material at the time was very nascent and very hard.
Right.
And now, we've got Moderna and BioNTech producing COVID vaccines in less than a year using, a related technology. And it's, something we saw coming 4 years ago. And we built a platform in-house to do precisely that, which we're very pleased with. And then beyond that, so we started building that, and we literally started with $50,000 from a friend of mine, fourth co-founder. He's not really co-founder, but he was like the first money in, right? Within days. Matt Cicero, who's been so supportive throughout. So it was us, 50K, and we were very lucky to be friends with a team called Corva Labs, who had some lab space out in San Dimas, and they happened to have a spare lab room with a steel bench in it, and it was a couple of 100 square feet. And we spent a year working out of it, and that's where we developed the first version of the technology and started getting proof of concept data. So really we had a lot of help early on, but we definitely took a longer road, and it took a long time. We hit it was much harder to develop the platform than we thought it would be. And I think anyone who starts a company, nobody really believes how hard it is, or nobody would ever do it. If it was a straightforward path, it would already have been done, right? So we worked on that, and that got us enough traction to attract the attention of a guy called Rob Reinhardt, who was the founder of Soylent, right? So this meal replacement drink that everybody lives on in the Bay Area. So Rob had done that. He built that company very well, and he decided to, all the innovation part of that was done, and now it's just a marketing problem to sell that product to as many people as possible. So he didn't want to be the CEO of that anymore. So he moved on then to angel investing. And he built this really cool space in downtown LA in a warehouse where there was an honest-to-God biotech lab and tissue culture facility that wasn't really operational, but it was there. And, we were introduced to him by Llewellyn Cox, who was the founder of Lab Launch, which is a network of incubator spaces in LA that he built from nothing, actually, which is in and of itself, that's a great story. And you should talk to Llewellyn in one of these.
Yeah.
So he'd introduced us to Rob, and we got to know Rob, and he had this vision, and he basically wanted to foster cool technologies. And it was a very eclectic space. There was like an artist's loft there as well. And there was a company that was called Ambicycle that was recycling clothing, and just very mixed bag of very much kind of an old school kind of, like where you would have like a sponsor. So he was like a patron of the arts and the sciences and all of that. And he had this cool space downtown, and we moved into that, and we were there for a good year in our first real biotech lab. And from that, we got, so this is actually a cool story. So we were in this lab working away on our platform, and we were still pitching Karma as we're the replacement for viral delivery systems, and getting nowhere really with investors, other than Rob, who saw, okay, like, yes, I see why that's needed. So I'm talking to Rob about something completely unrelated. He's looking at this investment, and it's a CRISPR company. And he was asking me about, what if the patient responds to the CRISPR, and that like with all gene therapies, and CRISPR is a bacterial protein. So he was worried that if you repeatedly dosed with CRISPR proteins, you would just get an immune response and it would stop working. And I think he's probably right about it. And he asked me this very naive question. He was like, why can't you just specifically turn off the anti-CRISPR immune response? And I said, well, okay, good idea, Rob, but think about it. If you could do that, you could cure every autoimmune disease, right? If that was possible, you've just solved every autoimmune disease. And he's like, tell me more. So I told him about this concept that I'd had, and I brought this up in the early days with Karma that I call Xavians. And that's just a cute word for a tolerogenic vaccine, which is a mouthful. So a traditional vaccine that we're all super familiar with, like the COVID vaccine, for example, teaches the immune system, this is the bad guy, activate and don't get it, attack, right? That's, and everybody's used, like when you think vaccine, you think take out bad guys. But that's not all the immune system does. Like we talked earlier about self versus non-self, right? The other mode that the immune system has is tolerate. And a vaccine doesn't necessarily have to be used to train for attack mode. A vaccine can also be used to train the immune system to tolerate. We call those tolerogenic vaccines, or reverse vaccines, or negative vaccines, which is a terrible, terrible phrase. You know, like vaccines have a bad rap already, and then you stick negative or reverse in front of it, and people are confused. And it just sounds
would you say it's so Zavines is more like a defensive vaccine?
Exactly.
And the typical vaccine is more offensive.
Yeah, right. Okay. So it's the flip side of the coin, right? So you know, Zavine is XAV, which is just vax flipped, right? So at the time it was sort of shorthand that we wrote on a whiteboard, and it stuck, you know. So we talk about Zavines all the time. We talk about Zavines. It's what Karma has become, because I was having this conversation with Rob, and he asked me, okay, how you've clearly thought about this, you know, how do you turn off specific immune responses? So I told him this story about, you know, the whole reason I wanted to do a gene therapy platform is because I wanted to develop a specific application using it, which I call Zavines, to solve autoimmunity. And he's like, oh, that sounds cool. Tell me more. So I launched into the story about my mom and how, you know, it's led me on this 30-year quest. And he's looking at me like I've grown an extra head. Right. I'm like, what? And he's like, why is that not your pitch? That's a great story. Like the tool thing, yes, it's important and I get it. But if you can pitch this story about why you're on this journey and why you're the person that wants to solve this problem, and then you've built this technology to enable you to do that, that is a fantastic story, and it's going to resonate with customers. And I think you'll have more success pitching that, even though you're building the same company, really. It's just how you present it, and the why matters, not just the what. And Rob, to his credit, that was the best piece of advice of many good pieces of advice that he gave me. And I changed that. I made a whole new pitch deck focused on that, started sending that around. And long story short, 2 months later, I was in a room with Vinod Khosla up on Sand Hill Road, and he's nodding, and his words were, "This looks like fun." And the next meeting was, you know, I think 2 meetings after that, we got our term sheet. So that's cool. That's basically the story of Karma and where we started.
Love it. So we have 9 minutes. I want to do some rapid fire style questions. So like 1 minute on each topic. What is Karma Biotech? It definitely sounds like a very good Karma.
Karma Biotech is developing vaccines for autoimmunity. So we are training the immune system to stop attacking the patient's body, which is the cause of autoimmunity. This has not been done so far, but it is definitely the way the industry is moving. We're seeing more and more competition, which is good. We like that because it validates us. But yeah, so Karma's ultimate mission is to be able to engineer immune responses as needed in vivo. Cool. Starting with autoimmune disease.
What is unique about Karma if you compare it with your competitors?
What's unique about Karma is the way we built the company. We built it as a true startup. So we built everything in-house from scratch. We haven't licensed any technologies from anybody else. We own it. We designed it. We understand it. We worked intimately with it. In the last 12 months, we've had about 500 iterations of the technology to settle on the one that works. And our technology is safe, scalable, and extremely flexible.
Cool. Do you have any patents?
Yep, I think we're about to get one awarded actually.
Very cool.
Soon. And we are filing new IP based on the last few months of work any day now. The things we've done in this last year have dramatically moved the technology just in terms of its capability, how unique it is. We've really done so much work, and that original IP, it looks like a very basic prototype compared to what we have now.
Right. You're creating a new class of a drug called Savines.
Yep.
What is it, and how is it different from current, like, other alternatives?
Yeah. So the problem with autoimmune diseases now and how we treat them is that we basically suppress the patient's entire immune system for life,
right?
Which is a very blunt tool. And it has all kinds of problems, not least, you're much more vulnerable to infections, right? So people like my mom cannot go outside during COVID at all. So she's being shut in for basically 10 months. You're more likely to get certain cancers. And then you get a lifetime of side effects from the drugs that are keeping you alive.
Right.
This is 20th century medicine. What Karma is doing is treating autoimmune diseases with 21st century medicine. So a Zavine is designed to just switch off the rogue autoimmune cells that are actually causing the problem and leave the rest of the immune system alone so that that patient can enjoy a relatively normal life and still be able to respond to things like the flu and COVID and all that stuff.
Love it. Love it. You are based in LA, Los Angeles. So
Yeah,
what is the biotech
LA.
scene? What is the biotech scene there? Because I was in San Diego. I know biotech is booming in San Diego.
Yeah, so LA is California's second largest hub for biotech now. People don't realize it is a little more spread out, but biotech is booming in LA. We have been an LA company from day one. We plan on staying that way. We're long LA. I built 2 biotech companies in LA, one about 7 years ago now when we started. There was nothing. There was no community. There was no VC. There were no labs. Very, very difficult. Now there's lab space. So Lab Launch is a great example. If you need incubator space, go to Lab Launch. There are VCs setting up offices here. Biocom, which is the biotech industry trade group, they set up an LA office, 4 years ago? Total game changer, complete game changer. Biocom have built basically the nexus that everything else is coming around. Steph Shi is in charge there now. She is a phenomenal ex-CEO of biotech herself. She gets it. She knows what we need. So it's absolutely the place to do biotech business right now. LA is biotech strong.
Like, why? Is it more affordable compared to, let's say, South San Francisco and Boston?
Yeah.
Or like, what is the reason why it is?
Right. Yeah. So there's a few reasons why LA is a good place to start biotech. Firstly, it is cheaper than, say, South San Francisco. A big thing that a lot of people don't realize is LA basin universities churn out more PhD scientists and engineers than anywhere else in the world. Right. And up until about 5 years ago, they've all gone to San Francisco or Boston or San Diego to go and get jobs. And that's changing now. And these really bright, young, especially in the biosciences, PhDs, they want to stay in LA. They like LA. They might have family here, or, they might have a partner that's still at school or whatever it is. Very hard to get jobs, right? Like, I built a company myself straight out of postdoc because it was hard to find the right fit for me. If it was now, maybe I would have joined a biotech and learned the trade a little bit, but it just wasn't a thing then. Now it is. One of the PhDs I hired for Karma, I hired her. Her name is Mariana Ochoa. I hired her right out of USC, right out of her PhD. Very bright, incredibly impressive. And she straight up told me in her interview, I want to start a biotech company someday, and I want to learn the ropes, and I want to join a growing biotech now. And when you've got people like that who want to stick around and they want to do what you're doing, right, and that's the next generation, and they're right here and want to learn from you, that is a great feeling, because I feel like the mentorship I got 5, 6, 7 years ago, I can pay forward now. And that's the big killer thing, I think. There's enough experience here, people who want to stick around in LA and mentor the next generation. It's a big deal.
I couldn't agree more. We need more bio entrepreneurs, and best employees are always want to start their own thing, because they look at things beyond day-to-day politics, right? They want to learn and grow. So, and we need more bio entrepreneurs. What advice would you give to aspiring bio entrepreneurs, like future Andrew?
There's a few things I would do. First of all, if you can, there's more and more entrepreneurship classes and courses and things that you can do. I'd recommend doing something like that. A really good one, if you can get in, it's very competitive, is the Keck Graduate Institute has a biotech boot camp, which lasts 2 weeks. They do it every year. I went to their first one, I think. Loved it, learned a lot, learned what I didn't know anything about, and started learning new stuff when I got home. Now I'm a panelist for that, so I've come full circle on that. And I see these really bright people from all over the country who want to learn the basics of industry and biotech entrepreneurship. So that would be a great place to start. They have a phenomenal program, something like that. If you can join a fledgling biotech company, you will see everything that happens there. With Karma, by necessity, we all share one big office. So the entire team is in there. We're all spread out in distance now, so we're a little further apart than we are normally. But my new hires see everything I'm doing. They hear every phone call. They see all my virtual pitches now, which is something that wouldn't have happened. A year ago, I'd have been off in San Francisco, and they wouldn't have been exposed to me pitching some VC somewhere. Now they see all of that, and I feel like if you can get into a company as an intern, or everyone needs help all the time. It's hard now because we have a maximum capacity that we're at, not because of the space we're in, but because of COVID, right? We just don't want as many bodies as we could potentially squeeze in. But at some point, having interns who are coming to the end of their PhD, or they're just wrapping up or whatever, I'm open to that. And I know a lot of my contemporaries are as well. That's a great place to learn. And then, as these companies grow, as we grow, when we know you and we know you're cool and you work hard, and you have good ideas and you contribute, you understand startup culture and you get it,
right? Then
you become an obvious hire, right? So that's what I would do. I'd try and get in to something like that. If you are starting a company, or you have started a company, one of the first steps I would take is try and get into an accelerator program. And the best one for biotech is IndieBio. So it'll mean moving to the Bay Area probably for 6 months, which is what I did.
Or New York.
Yeah. They've had their first cohort there. But IndieBio, they take 30 companies every 6 months or so, invest a couple $100,000, give you some lab space, and then more importantly, train you how to pitch, how to talk about your company, how to find that product market fit, and put you in touch with this huge network of investors. And that is, it's an accelerator, and it really does accelerate the progress of your company. And SOSV and IndieBio, SOSV is the venture fund that backs IndieBio. They've been so supportive, brilliant collaborators, and I would strongly recommend trying to get into that program. Very hard. And I guarantee you your first pitch deck that you put together for them, if you show it to me first, I will put a lot of red ink on it. But, if you can get into a program like that, it will really accelerate.
Great advice. So Andrew, thank you so much for joining this episode of
Focus
Biotech.
It's been fun.
Lot of good for Karma. I can tell you that.
Yeah. Thank you.
Talk to you soon. Thank you so much.
Yeah. Thanks, Guru.
Thank you. Bye.
Bye.